Now let's look at the enzymes and certain metabolic pathways in this section. What is a metabolic pathway? A metabolic pathway is a series of reactions that will start. For example, if I have a molecule A and a molecule A converts to B and B converts to C and C to D, and eventually you might get E, and this might be your final product. So a series of reaction in which one molecule helps the formation of the next molecule is metabolic pathways. Within our cells, most reactions are carried on by metabolic pathways, which means that no reaction is just a one-step reaction. It's going to be a series of reactions. Now, it gets more complex when you look at all the reactions that are going on within the cell, that each reaction is then going to be controlled by its own unique enzyme. So let's call this E1, and this could be E2, and this could be E3, and this reaction is E4. So if there are a series of 20 reactions, you will find that there are 20 different enzymes that help catalyze this reaction. Now, when we say the word catalyst, what is a catalyst? A catalyst is a substance which is usually, when we use the word catalyst, we're not just talking about enzymes. It could be temperature, it could be a mechanical agent, it could be a metal ion. What a catalyst does is it lowers the activation energy. So what it is enzymes lowers the activation energy for any kind of cell, any kind of reaction. Now from this chart, you can see that this is the free energy, which is on y-axis, and notice that for a given reaction, the energy requirement would be as high up as up here. Now, if an enzyme is present in the same reaction, the reaction can take place at a lower energy level, and this is what we talk about lowering the activation energy. Now, when we use the word catalyst in other classes, a catalyst could be heat. Now, you cannot really take heat and start heating up your cells to release the energy that is stored in glucose, because the temperature would probably destroy the macromolecules in your cells. So, biological reactions are catalyzed by enzymes, and all enzymes are protein in nature. So here we have a massive group of substance, which is the enzymes, and these are proteins in nature. And each enzyme will work towards its own unique substrate, which means that each enzyme recognizes a molecule that it will work on, and that molecule that it works on is called as a substrate. And the name of the enzymes usually will end in the suffix ase, but not always the case, but most of the time you will find the word ase. For example, sucrose is broken down by sucrase, and there's lipase, and most of them will end in ase. But again, I'm not saying all, because when you'll go into anatomy and physiology, you will find trypsin and pepsin, which are not ending in ase, but most of the time it will end in the ase. So if you have a reaction that has got 20 steps, there are possibly 20 different enzymes that are catalyzing each of these reactions. So now let's see how do the enzymes work. Enzymes are very unique, that enzymes have a region which is called as the active site. The active site is mainly a chemical configuration of the molecule. Now remember, these are proteins. We discussed proteins earlier, and you know that they're made up of chemical bonds. So a unique structure of a bond will give this unique shape, which is the active site. Now when an active site is available, then this active site is now going to look for all kinds of substrate to work on, and the true substrate would be the one that can fit into this active site and form new bonds with the enzyme. When these new bonds are formed, some of the old when the new bonds are formed, linkages between the substrate and the enzyme, that helps break some of the older bonds within the substrate, and eventually the substrate can then release new products based on these new structures that are forming on new linkages. So here in this example, we can see that this is an example of synthesis of molecules, and notice over here. Okay. Now in this synthesis reaction, notice that we have the enzyme over here that has its own unique shape, and they're shown different substrates of different configuration. And the true substrate would be the one that can fit into this this active site or the spatial configuration, and eventually make new bonds and reconfigure to form the product. However, the enzyme is released in this process and can go back and pick up a second molecule of substrate. In the reaction on this kind, which is a degradation reaction, which means breaking up of a larger molecule to smaller products, the substrate is fitting into the active site, forming a complex, and eventually splitting the two substrate bonds separately, and you can see new products that are forming. Now this the models that I'm just explained to you is sometimes called as the lock and key model. The lock and key model corresponds to having your lock and your key, lock of the car, and the key that you open up with. So if you have the right key, you're able to open up your car or any kind of lock. So it's the right configuration. So even though I might have my own key, but I can't go ahead and open all your cars because it will not fit in there properly. The second concept is that sometimes enzymes work with the induced fit model. The induced fit model explains that when the active site is combined to the substrate, it induces certain alterations in the shape of the substrate, and this the change in the active site then eventually helps the substrate molecule to either bind to another substrate or to break apart. I think of it as a pair of hands. If you have a pair of gloves and you try to put your right hand in a left hand glove, you can see that it does not fit in properly. But if you fit your right hand in a right hand glove, it causes a nice fit, and it helps the molecules expand. That is the concept of the induced fit. Induced fit is more of a 3D conceptual to understand. Lock and key is more 2D, as shown over here in this example. So both of these models are there. Your book mainly mentions the induced fit model, but I went ahead and explained to you the lock and key model because you might hear of that in some other class later on. Enzymes are protein in nature. So since they are protein in nature, there are various physical factors that can affect its rate of reaction, which is how fast or how slow will it work, and that is that energy of activation is either increased or decreased by any of these factors. So the first one we will look at is called as the substrate concentration. The substrate concentration means that the substrate molecules that are available to the enzyme. If you have more substrate available, because there as if there are more substrate available, and as the enzymes releases its active site, more products can be formed. Does that make sense? So because if you have only 10 substrate molecules, then you'll only have 10 products. But if you have 20 substrate molecules and you only need maybe perhaps 3 molecules of enzymes, you will speed up the reaction because you will have more products forming. So but again, if you add 100 substrate molecule, have you really increased the reaction? Not really, because it will come to a saturation. So if I was to do a graph like this, in which I have the substrate concentration on this side and the reaction rate on this side, then most likely I will get a plateau, because even if you keep on increasing the number of substrate, if you only have a limited number of enzyme, it is not really going to dramatically increase your rate of reaction because only a certain number of products can be formed. Let's look at the temperature. The temperature is shown over here. You can see what happens with temperature, that as you increase the temperature, enzyme activity increases. It reaches a maximum or optimum, and then it declines over there. Now, if I was to take the range over here and look at the range that we have, my optimum temperature, notice it's around 37 degrees Celsius. 37 degrees Celsius is the body temperature. So that the enzyme activity will be maximum in that temperature range. If you increase the temperature, the enzymes are all proteins and they will break down. So it will mainly destroy the enzyme. Again, if you keep it in cold, for example, if you want our food not to get spoiled quickly at room temperature and we leave it in the cold temperatures, then it slows down the enzyme activity. Let's look at the next physical factor, which is pH. pH for enzymes are very unique. Certain enzymes work at acidic pH, as you can see over here, and certain enzymes work at alkaline pH, as seen over on for trypsin. Again, there is a range for the enzyme activity. The curve is the similar pattern as we saw in temperature. However, the enzymes have a very narrow range of resisting pH. So if you have a drastic change in pH, unless you have buffers in the system that will resist the pH change, enzyme activity would be either declined or at extreme ends of the pH. Another factor that is needed is called as the cofactors and the coenzymes, which is the amount of enzyme that is present in a system is always regulated by certain cofactors. Cofactors are usually inorganic ions. So if we take cofactors, for example, calcium ion would be a cofactor, iron would be a cofactor, zinc ions would be cofactors. So usually cofactors are going to be inorganic ion or a non-protein organic molecule, and these are usually the non-protein organic molecules are the coenzymes. So let's call these non-protein molecules. An example of this would be vitamins. Vitamins would be considered as the coenzymes because they are not protein in nature, but they are organic molecules. They're small organic molecules, and both cofactors and coenzymes are usually needed in very trace amount, and that's why the vitamins are essential to our diet, because trace amounts of these are will help the enzyme to work in its optimum rate. How do these work to get to the optimum rate is by a process called as phosphorylation. Phosphorylation means adding phosphate to a molecule. So enzymes get activated when they get to activate a protein. For example, over here you can see a certain amount of kinases, which are enzymes, are activating a protein, which is shown in this part, which is the inactive molecule. And when the kinases help to attach the phosphate group, the molecule is more active and it can then undergo certain reactions. A lot of hormones work in this way. They might be present and they might be in an inactive state, and when they get phosphorylated, then they become active hormones. We looked at previously all the different factors that help increase enzyme activity. Let's look at a reaction that actually slows down an enzyme activity. Enzyme activity can be stopped by a process which is called as inhibition. Inhibition means slowing down, or sometimes it actually stops. It's an imperative path. It is imperative for any pathway to be coordinated and regulated. Enzyme inhibition occurs when a substance known as an inhibitor will either slow down the reaction by inhibiting to the enzyme, or it will decrease its activity. There are two kinds of inhibition. One is called as a competitive inhibition, and the other one is called as a non-competitive inhibition. So let's look at each of them. In a competitive inhibition, the substrate and the inhibitor are both able to bind to the enzyme's active site. For example, when you get sick and you're given antibiotics, then the antibiotics is designed to be similar in shape to the substrate molecules of the bacteria. So the substrate of the bacteria and the antibiotics are both competing for the enzyme, and each will try to form its complex. Once the enzyme is able to bond with the bacterial with the antibiotic, then it forms an enzyme substrate complex, and because it's not the true substrate, it actually starts slowing down or inhibiting this reaction. So for example, if we have enzyme plus the substrate forming an enzyme substrate complex, and this complex is not the true complex for the bacterial activity, it will eventually slow down the bacterial activity and the bacteria will die. This is called as a competitive inhibition, where the substrate can be either an antibiotic, or it can be the true bacterial substrate. And that is why when you don't finish up or people don't finish up their antibiotics, the bacteria is able to come back, is because it's competing for the active site. And when there's no more antibiotic available, then the true bacterial substrate is available for the enzyme, and it starts forming the enzyme substrate complex, and the reaction can then go on and form its correct product. On the other hand, a non-competitive inhibition is the one in which the enzyme binds to a part of the substrate binds to a part of the enzyme, and it totally inhibits the way. The diagram for that actually is over here, which is the inhibited pathway. And in this, examples of non-competitive inhibition are, for example, poison of all sorts. When you have, or when a cell is injected with any kind of poison, then the inhibitor binds to the enzyme at a location other than the active site, and that other site is usually the allosteric site. And this will eventually slow down, or actually it will just end the reaction. So poisons would be examples of non-competitive inhibition, and antibiotics would be examples of competitive inhibition. The top reaction over here is your example of non-competitive inhibition. So if you can relate to each with a common example, it will help you remember as to which one is which. Enzyme inhibition of the competitive kind is usually reversible, which is this kind. You can usually reverse it with either increasing or decreasing the number of the substrate molecules, the true substrate, or the substrate that you're trying to fool it with. Our next section is about oxidation reduction reactions. We'll hear a lot of